But the most important thing is once you validate your assay and you check that the reproducibility is good using a series of technical replicates and things like that, known standards, that's the most important part to get a good, robust, reproducible assay with small variation.
Hey, smart scientists. Welcome to another episode on the Smart Biotech Scientist podcast. If you have tuned into a few episodes on this podcast, it may not come as a surprise if I tell you that I'm passionate about glycosylation. And that's why I'm excited, because today I'm having a conversation with Róisín O'Flaherty, who is an assistant professor in the Department of Chemistry at Maynooth University in Ireland. And we are talking about glycoanalytics and glycoengineering. Are you juggling the complexities of CMC development while trying to enjoy the beauty of biotech? Have you ever wondered if there's a way to simplify bioprocessing? Welcome to the Smart Biotech Scientist podcast, where we're diving headfirst into the very challenges you face. We're breaking it down, demystifying the jargon, and giving you the keys to unlock your full potential. I'm your host, David Brohlman, and I get it. With 15 plus years in the biotech industry, I face the same challenges you do. There's a way to simplify and streamline so you can remove complexity, you can skip trials and errors, deliver without delay your groundbreaking therapy to clinics at market, and still enjoy every single step. Do you want to learn how industry experts and I did it? Grab a cup of coffee and your favorite notebook and pen. Now is the time to take your bioprocessing game to the next level. Let's smarten up biotech. Welcome, Róisín, to the Smart Biotech Scientist podcast.
Thanks a million. It's lovely to be here.
It's a pleasure to have you on the show today, Róisín. Share something that most people don't know about you.
Okay, so I suppose because I'm working in the biopharma industry, I guess there would be an assumption that I have studied biology, but I have never formally studied biology. So I did chemistry, I did physics, mathematical physics and mathematics, and I actually thought I was going to be a physicist at the beginning, but then ended up loving chemistry, so went into chemistry. Then went into engineering, and after engineering went into biology. So I guess that's a little bit of a different background for someone working in the biopharma sector.
That's an exciting story. What motivated you to enter the biotech industry, and what led you to your current position in academia?
So I think I did my PhD in glycochemistry. So like some people call it glycochemistry, some people call it carbohydrate chemistry. But it was very much organic synthesis. And that would have been my first love when I was doing that. But I remember when I was doing my PhD, going to a talk by Carolyn Bertozzi. At the time, she was visiting Ireland. She's a Nobel laureate and a really great glycoscientist. Her work was kind of on that interface between chemistry and biology. And I remember being really, really inspired at the time. So when I finished my PhD, I also, as I kind of mentioned, have this love of engineering and mathematics. So I went to Intel semiconductor industry and I was working there as an engineer for a few years, but I really missed the research end of it. So then a job post came up in a place called NIBRT, the National Institute for Bioprocessing Research and Training. And they wanted somebody with an interest in robotics and somebody with an interest in glycoscience. And I actually had the two of them, albeit from different sources. So that's kind of where I came to start getting exposed to some of the biological aspects, I suppose. But really, I was a trained chemist with a background in engineering. And I just kind of learned the trade while I was there. So I started off working for a PI called Pauline Rudd. She's really well known in the glycoscience communities and also in the biopharma sector. And then I transitioned into cell technology then when I moved into the lab working as a postdoc with Mike Butler, again working in cell engineering. So it just kind of naturally happened based on the background and expertise that I had. It was just serendipitous in some ways.
You come from a non-traditional path and from a totally different side into our industry. What do you think are the main challenges that biotech scientists often encounter in the bioprocess development and in the manufacturing of biologics?
I think everyone's probably going to give a different perspective here, but probably for me, the biggest challenge is probably reproducibility and the challenges that that then presents. So whether you're working in academia or whether you're working in industry, I think that's personally the biggest hurdle, because obviously if you want to keep your product within tight controls or specifications, any subtle changes that you have where you don't have good reproducibility or you have high variability has a bit of a knock-on effect. So you have the butterfly effect, where if you have a 20-step process and in the first step you have this large variation, you can imagine then as you're going on in your process that the gap becomes wider and wider. So personally, I think everything where you can automate, I would automate where possible, or having exact conditions as much as you possibly can reduces that margin of error, I suppose.
Now let's move into the main topic of our conversation, glycoanalytics. Can you give us a simplified overview of glycoanalytics and also explain why this matters for the biopharmaceutical industry?
Okay, so glycoanalytics is the study of glycosylation or glycans or sugars. So sugars are known as being a critical quality attribute of biopharmaceutical products. So the monoclonal antibodies or your fusion proteins. And the reason that they're a critical quality attribute is because they really influence things like the function and structure of your protein products. And glycans, it's a fairly diverse term, so you can have lots of different types of sugars, but the bigger two classes are sugars called N-glycans and O-glycans. And critically, depending on which type of sugars you have, these can be immunogenic, so they can be harmful to humans, but also they can be incredibly powerful. By switching out the sugars that you have on your proteins, you can increase its efficacy, the clinical efficacy. You can increase its half-life, so how long it's going to stay in the body. So if you have knowledge about the glycosylation or the sugars and studying them using techniques like glycoanalytics, that allows us to fine-tune our biopharmaceutical drugs and use more lower doses and be safer for humans.
And now a lot of things have happened in this space. What are the most prevalent techniques we have at our disposal today?
Yeah, so I suppose broadly speaking, you've three different major groupings if we want to think about studying glycosylation. And specifically, if we're talking about glycoproteins, it changes a little bit if you're studying glycolipids or other things, but the vast majority of biopharma, they're based around glycoprotein structures. So you can do three different approaches. You can study the glycoprotein in its native form, and traditionally we use mass spec type approaches for that. And that presents with lots of challenges, but also lots of advantages. It's relatively simple, but some of the information that we get mightn't be as detailed as we might like. But again, there are lots of advances in that area. The second one is where you use enzymes like trypsin, things like that, which break down the glycoprotein into smaller fragments. So this would be glycopeptide analysis. And predominantly we use mass spec-based approaches when we study glycopeptides, using predominantly, again, reverse phase technologies. But again, the scope is there. And then the last grouping is where you clip it even further and you clip off the sugars. So you break down the glycoprotein into its glycopeptide. And then what you do is you use enzymes to clip off the sugars and you study just the released sugars. And that gives us the widest amount of information. And historically, if you think about the development, this is where we probably would have started off in the 80s, clipping off the sugars using this released glycan approach. But again, you can also look at combinations of the three and merge it all together. And they each give us little pieces of the puzzle to kind of piece it all together. So there's the three broad ways that we can analyze glycosylation.
Some of our listeners are working in the production of recombinant proteins. And from my experience, having worked in that space for many years, you face different challenges. So how do we select the different methods depending on whether we are in a high throughput setting, for instance, during process development, or doing a structural analysis of our molecule?
You dramatically change what approaches that you have. So if you're doing QC assays, you want to go with the simplest, most cost-effective method, which shows and highlight some of the key attributes that you're looking for. So if you know you're producing your product in mammalian cell culture, you might have to look for immunogenic epitopes like alpha-gal or certain sialic acid motifs. So if your technology can convincingly show the regulators that it's reproducible using your assay, then that's perfect. So for example, you might in that case just use native glycoprotein analysis where you can show that you're reproducibly getting the same assay. Or you could use your released end glycan approach and again show the reproducibility. So you're just running the sample through the system. However, if it's completely unknown, then you have to do far more of a kind of deep dive and it takes longer. So you can either use mass spectrometry where you feed it into the software. You can use a targeted approach where you're going after certain ions that you know are indicative of glycans. There are databases that you can cross-reference against. And then the other thing that we do a lot in our group for unknown samples is we use enzymes that are known to clip off certain sugars. So we'll analyze the glycans, then we'll clip off the sugars, and then we'll reanalyze them. And by comparing the differences of the profiles, we're able to identify what glycans and what linkage type they have. We can tell an awful lot, and then we combine that with mass spec. So for unknown samples, it could, depending on the level of detail in the sample that you're studying, it could take 3-4 months for the first time that you're studying a new mechanism. But if it's something simple that you've already characterized, then it's a case of a 5-minute run through your protocol. So it really depends on your application. But at the beginning, it takes time to set up your assay. But once it's set up, and we're big on automation in our group, we've already run 10,000 samples and more through our automated systems so that we have this high throughput platforms.
What is the typical workflow for these methods?
So the most important thing is sample prep. And again, this sample prep really depends on the complexity of your sample. So if you have something coming from cell culture, you just want to do some cleanup steps, which gets rid of the cell debris. If you're going after particular glycoproteins, then you might use affinity chromatography with something like protein A or protein G, or you can do size exclusion and all those other things. But that is the most critical step, is that sample preparation. Because you know that whole concept of shit in, shit out. If the data is good to begin with, then we're going to get much more usable real data at the end. That is, for me, the most important part is sample prep. Then you use your technology. So if you're interested in glycopeptide site analysis, then we'll use mass spectrometry. Or if you're interested in released glycan, then what we do is LC or UPLC based approaches. And then you just quantify your glycans at the end. But the most important thing is once you kind of validate your assay and you check that the reproducibility is good using a series of technical replicates and things like that, known standards, that's the most important part to get a good, robust, reproducible assay with small variation.
What I'm hearing, Róisín, is that there are a lot of moving parts you have to manage. What role does automation play here? And how does it augment these approaches and workflows?
So for us, we have developed fully automated technologies, except for when we are quantifying at the end. So the most timely part of our workflows is actually the data interpretation. So in my opinion, that's where we need the largest amount of kind of concerted efforts. And to that end, tools like bioinformatic tools or system biology approaches, AI, I think are going to be incredibly insightful and important for us. And, you know, there are a lot of players in that field that are doing really good work, like Daniel Bojar, Lisa Fada, Nathan Lewis. Some of these people, they are really bringing us forward, particularly studying glycans, because as you're probably aware, David, it lags behind other fields because I think people don't have the same confidence when they go to study glycans because it's a little bit, I wouldn't say more complicated, but it's not as template driven, let's say, as if you're studying proteomics or genomics. And so people shy away from it. But the more tools that we give to make it more user friendly and more databases that we can use, it's going to help us all in the long term. And it'll mean we don't have to manually go back to these mass specs and search manually for these glycans. Hopefully we'll have these tools that will just feed in our data and we'll just be able to interpret the results.
Róisín, considering the emergence of many new technologies in these exciting times, where do you see this going? For instance, with advancements happening daily, particularly in AI, what do you anticipate will change if we were to have this conversation 5 years from now?
I think the trend is that, so traditionally, 5 years ago, even 10 years ago, I would say the vast majority of people were studying their glycans by released glycan approaches. So whether that's released N glycans or released O glycans, the vast majority of scientists were using that technique. Now I can see the trend in academia and industry is there really is a shift towards glycopeptide analysis, which is mass spec. But mass spec is pretty expensive, right? So it's not accessible for every scientist. So I think there will always be room for other technologies to accommodate your particular scenario. But for sure, I was involved in a study performed by the National Institute for Standards and Technologies, NIST, where they got 100 labs to analyse the glycosylation of these unknown biotherapeutics. And what that study showed was that there's an increased prevalence in using mass spec, not only in R&D labs, but also in routine manufacturing. And so I think that will increase. And particularly, I think there's a lot of interest lately in ion mobility and how that might open up. So I think possibly in 5 years' time, we're going to be seeing more glycopeptide analysis and using mass spec.
Hey, smart biotech scientists. I hope you've enjoyed our conversation with Róisín O'Flaherty on Glycoanalytics. Róisín O'Flaherty is an assistant professor in the Department of Chemistry at Maynooth University in Ireland. And coming up in part 2, we are talking about glycoengineering. So stay tuned for an insightful conversation. And perhaps you would like to develop a robust and cost-efficient manufacturing process, but you feel overwhelmed and you don't really know where to start. Don't worry, because we would like to help you get started, and my offer is very simple to you. Just book a free call using the show notes below, and we'll help you make an action plan so that you can develop biologics with peace of mind. All right, smart scientists. That's all for today on the Smart Biotech Scientist podcast. Thank you for tuning in and joining us on your journey to bioprocess mastery. If you enjoyed this episode, please leave a review on Apple Podcasts or your favorite podcast platform. By doing so, we can empower more scientists like you. For additional bioprocessing tips, visit us at smartbiotechscientist.com. Stay tuned for more inspiring biotech insights in our next episode. Until then, let's continue to smarten up biotech.